Segmentation in Component Analyzer. In this video, we demonstrate the latest functions in FluoRender's Component Analyzer for segmenting structures. Open an example dataset called dens_field.tif. This dataset contains several round structures need to be separated. However, the difference of their intensity values makes the separation impossible using a single threshold value. We introduced a series of new functions to handle more sophisticated structural segmentations. First, an auto-update mode allows changes to the component generation settings to be applied immediately. Click the Auto-Update button to enable this mode. Change the settings such as iterations and threshold to see the result immediately. We see that when the threshold value is lowered, more blobs are identified. However, blobs with higher intensity values start to merge as well. We can find a threshold value just before blobs start merging. Intuitively, if we could remember the current state and then keep lowering the threshold without influencing the existing segmentation, the difficulty of uneven intensity distribution would be more easily managed. The Fixate Grown Regions function does that exactly. Check the option and the colors of existing segmented regions change, indicating that they have been remembered. Then, lowering the threshold value will not make remembered regions merge again. This process can be repeated by clicking the Refix button. Then, lower the threshold value again until the blobs with the lowest intensity values are segmented. Click Refix once more to finalize the segmentation result. Even structures with the lowest intensity values can be segmented easily using this method. To confirm the segmented result, go to the Analysis panel and click Random Colors to convert components to channels. Once the channels are generated, their colors can be more easily customized for a clear visualization. Right-click the group and choose Randomize Colors to assign random colors to the segmented channels. Alternatively, we can use the density field function to handle segmentation of data with uneven intensity distributions. Check Use Density field to enable this function. This function examines intensity values locally instead of globally. It divides the data into smaller regions. A local intensity distribution is computed for each region. Then, the segmentation is computed based on the intensity value relative to the local distribution. Therefore, a very low global threshold value can be used. Let's look at the density field settings next. The density control is a local threshold value, which controls how structures are separated. The window size value sets a range to smooth local intensity values. Set it to a small value only when small details need to be considered. The stat size determines how the dataset is divided. It is the size of each region that the statistics on intensity distribution is computed. Using the density field feature can identify the local peaks of intensity. To further grow the intensity peaks, use the cleanup functions. Check Cleanup to enable this function. Notice that the expanded regions will not merge with each other. Click Clean More to further expand each region until convergence. Similarly, go to the Analysis panel to export the components as channels for more clear visualizations. Another more difficult case for segmentation is when structures are connected by high-intensity values. Open the example dataset called diced_field.tif. It is impossible to segment this dataset by only intensity-based method. Therefore, we added a new function called the distance field. Check Use Distance field to enable this function. The distance field is built by successively reducing the intensity values from an initial outer perimeter. Then, The separation of the structures can be observed in the distance field. The strength adjusts the influence of the distance field. Then, we can use the existing segmentation functions and apply them to the distance field. The global threshold value reveals the distance field at different distance values. The threshold value of the distance field determines where the initial perimeter starts. Use a high threshold value to better separate the structures. The density field functions can be used along to equalize the intensity variations. Choose a density control value to separate the peaks of the distance field first. Use the cleanup function to expand the separated peaks without merging. The shapes of each separated region can be fine-tuned using the stat size setting. Notice that the result may be sensitive to how the dataset is divided for computing the statistics on the intensity distributions. Use Clean More to expand the regions if necessary. When a satisfactory result is generated, use the channel export function to generate new channel from the components. Alternatively, if finding a single set of settings to segment the dataset is too tricky, use the fixate functions to successively segment the data. For example, we can only find settings that separate the high-intensity regions of this dataset by trying different combinations. We can fixate the high-intensity regions first, and then try to adjust the settings to bring out the low-intensity regions. Fixate the low-intensity regions again to finalize the result. In conclusion, the newly introduced functions in FluoRender's component analyzer, including the density field, distance field, ground region fixate, and auto-update, enable more flexible and higher quality segmentation for fluorescence data.